Insights, Basics and Beyond. Chapter 15. The Complement System. Marcello here. Welcome back to Immunology Insights, Basics and Beyond. In our last chapter, we discussed immune cell markers and receptors, the critical molecular components that allow immune cells to recognize pathogens, communicate, and carry out their functions. In this chapter, we will cover the complement system, a key component of the innate immune response that plays a vital role in defending the body against pathogens. So let's talk science. To introduce the complement system, it is a complex network of proteins that work together to enhance the ability of the immune system to fight infections. It is part of the innate immune system, meaning it provides a first line of defense against pathogens without requiring prior exposure or specific recognition by the adaptive immune system. The complement system is named like this because it complements, so enhances the immune response. Its proteins are labeled with a capital C for complement and numbers, for example C1, C3 and C5. When activated, these proteins are cleaved into fragments, usually designated with a lowercase a, which is a small fragment, often pro-inflammatory, and a lowercase b, which is a larger fragment, usually binding to surfaces or forming enzymatic complexes. Some of these fragments, for example C4b or C3b, help form enzyme complexes like C3 and C5 convertases, which amplify complement activation. While A fragments act as inflammatory signaling molecules, B fragments bind to surfaces for opsonization or enzyme complex formation, ultimately enhancing immune responses and pathogen destruction. The complement system has three primary functions. First, opsonization, so marking pathogens for destruction by phagocytosis. Second, chemotaxis, attracting immune cells to the site of infection. And third, lysis, directly killing pathogens by forming pores in their membranes. The complement system is composed of more than 30 proteins, most of which are produced by the liver and circulate in the blood in an inactive form. Upon activation, these proteins interact in a highly regulated cascade, amplifying the immune response. Let's now look at the activation of the complement system. The complement system can be activated through three distinct pathways, each triggered by different signals but converging on the same central component C3, which plays a key role in the system's function. We have the classical pathway. The classical pathway is initiated when antibodies bind to antigens on the surface of pathogens, forming immune complexes. This binding activates the C1 complex, which then triggers a cascade of reactions leading to the activation of C3. This pathway links the innate and adaptive immune systems by requiring antibodies produced by B cells. Then we have the lectin pathway. The lectin pathway is activated when mannose-binding lectin, or MBL, a protein that recognizes specific carbohydrate patterns on the surface of pathogen, binds to these carbohydrates. This binding activates MBL-associated serine proteases, or MASPs, which in turn activate C3. The lectin pathway does not require antibodies, making it part of the innate immune system's immediate response to infection. And last but not least, we have the alternative pathway. The alternative pathway is activated directly on the surface of pathogens, without the need for antibodies or lectins. In this pathway, C3 undergoes spontaneous hydrolysis, forming C3b, which can bind to the surface of pathogens. If not regulated by complement inhibitors, this leads to the formation of a complex that activates more C3, amplifying the response. Once C3 is activated, it is cleaved into C3a and C3b. C3b acts as an opsonin, marking pathogens for destruction by phagocytes, while C3a acts as a chemoattractant, recruiting immune cells to the site of infection. Next, let's look at the effector functions of the complement system. The activation of the complement system leads to several important immune functions. First, we have opsonization. C3b binds to the surface of pathogens, tagging them for recognition by phagocytes, such as macrophages or neutrophils. This enhances the efficiency of phagocytosis, allowing the immune system to clear infections more effectively. Second, we have the membrane attack complex, or MEC formation. The terminal pathway of the complement system involves the formation of the membrane attack complex MEC. The MEC is a cylindrical structure composed of the complement proteins C5b, C6, C7, C8, and multiple C9 molecules. This complex inserts into the membrane of the pathogen, creating pores that lead to cell lysis and death. The MEC is particularly effective against gram-negative bacteria and other pathogens with vulnerable membranes. Third, we have inflammation. Complement proteins such as C3a and C5a act as anaphylatoxins, which promote inflammation by increasing vascular permeability and recruiting immune cells to the site of infection. This helps to concentrate the immune response to the site of infection, facilitating the elimination of the pathogen. Fourth, we have the clearance of immune complexes. The complement system also plays a role in clearing immune complexes, which means aggregates of antigens and antibodies, from the circulation. This prevents these complexes from depositing in tissues, where they could cause inflammation and tissue damage. Let's also look at the regulation of the complement system. Given its potent effects, the complement system is tightly regulated to prevent damage to host tissues. Several complement regulatory proteins are present on the surface of host cells to protect them from complement-mediated lysis. These include the decay accelerating factor DAF. DAF prevents the formation of the C3 convertase, a critical enzyme in the complement cascade, thereby inhibiting the activation of the complement system on host cells. Then we have the factor H. Factor H regulates the alternative pathway by promoting the degradation of C3b and preventing the formation of the C3 convertase. Last but not least, we have CD59. CD59 inhibits the formation of the MEC on host cell membranes, protecting them from complement-mediated lysis. Deficiencies or dysfunctions in these regulatory proteins can lead to complement-related diseases, such as paroxysmal nocturnal hemoglobinuria or PNH, where red blood cells are lysed due to uncontrolled complement activation. Of course, with all its important functions, there are clinical implications and therapeutic applications for the complement system. The complement system plays a role in a variety of diseases, including autoimmune disorders, infections, and even age-related conditions such as macular degeneration. Therapeutic strategies that target the complement system are being developed to treat these conditions. One example is the use of complement inhibitors, such as eculizumab, which is used to treat PNH and atypical hemolytic uremic syndrome, or AHUS. These drugs work by blocking the activation of the complement system, thereby preventing damage to host tissues. In addition, research is ongoing to develop vaccines that enhance the complement response against pathogens, as well as therapies that modulate the complement system to improve outcomes in diseases like Alzheimer's disease and cancer. To conclude, the complement system is a powerful and essential component of the immune response, playing a critical role in defending the body against infections and in maintaining immune homeostasis. Its ability to enhance phagocytosis, promote inflammation, and directly kill pathogens makes it a key player in the innate immune system. In our next chapter, we will go more into detail of the major histocompatibility complex MHC that I briefly already introduced in a previous chapter. However, its importance necessitates a separate chapter. MHCs are a group of molecules that are crucial for antigen presentation and the adaptive immune response. So stay with us as we continue to delve into the fascinating world of immunology. Before closing, I have a small request. If you enjoyed my content and want to hear more, like and subscribe. It's a small step for you, but it helps me a lot. So see you next time and thank you for listening.